talk about identifying L1029H mutation against permethrin pesticides in North Carolina tobacco budworms.
All right. So, like Megan just said, I've been working on a project at NC State University with my mentor, Dr. Fred Gould, and Miss Alex Yankee. All right. So, how we counter resistance today determines what we eat tomorrow, if we eat tomorrow. And what I mean by that is that resistance against pesticides has become a recurring problem in agriculture today. And so basically, the resistance goes through this cycle of farmer spraying the pesticide on the crop, and as you can see through this demonstration, that it kills off every susceptible population in the area, but it leaves a majority of the resistant population to continue reproducing, and then just the next generation tends to be filled with resistant insects, and basically just renders the pesticide useless. And as the pesticide becomes useless, a lot of funding needs to be poured down the drain into funding research to find that genetic strain that is causing that resistance, and then developing a new pesticide or a GMO, genetically modified organism, that will counter the resistance, and then applying that to the field. But as more farmers continue to not really learn from what they've done before, the cycle just continues to repeat. And so, here are just like some effects of what that process looks like. As you can see here, we have insects eating tobacco crops, which are a main cash crop here in North Carolina. Crickets destroying a cornfield, and even a photo of what we encountered over the summer when we were collecting with a bunch of insects on tobacco flowers. All right, and so our goal was to be able to determine the genetic sequence in tobacco budworms that is causing this inherited resistance. And we went about that by collecting different insects from different fields all across North Carolina. And our hypothesis is that since these insects are becoming resistant to permethrin pesticide, that it would be the L1029H mutation, which has been a historic mutation that has been seen before as a citation, since it is becoming resistant against the sodium channels. So, what we did to kind of go about this experiment is that, like I said earlier, we collected samples from different fields throughout the summer. We went all across North Carolina, from Rocky Mount to Reedsville, even to the coast of Washington for our samples. And so, as we saw those samples to hatching, we placed them on a special diet which had a small portion of the pesticide. And with that pesticide, we kind of raised the insects, and as they died in the pesticide or survived, we cataloged them into resistant or non-resistant, so that's phenotyping and then genotyping. So then with that, we were able to isolate the insects into different groups and perform a RFLP assay. And so, like I said earlier, we collected samples, but we didn't collect the actual insects. We collected these eggs, as you can see in this image. These are images under a microscope. So, if you would like to imagine what we went through all summer, scavenging huge tobacco fields for these little eggs under the each little leaf. And if you want to imagine the size of them, they're about the head of a pin needle. So, that was fun. Next slide. Okay, and so as they hatch, we place them into these little containers that had a very small portion of diet which contained pesticide. And as the insects grew and died or survived for a long time on these diets, we were able to perform DNA isolations on them to be able to see whether or not, or to be able to see which specific strand that was present in their genome that was not present in the other one. So if they were resistant, we wanted to see which strand was causing the resistance that wasn't in the susceptible insect. All right, and so next slide. As we did this, we performed an RFLP assay, which was just a specific enzyme that went through the code that was in the genome, and it only cut whenever it saw the strand that we were looking for. And so, as you can see there, it had two key strands, which was susceptible sample. So it didn't cut anywhere, leaving only one strand of the susceptible sample. Whenever one strand of the resistant allele, it cuts there, and we see two strands, and same thing with the homogeneous resistant one, it cuts both the strands, and we see one band for the resistant one. And then you can see here, through a gel electrophoresis, that we kind of ran the samples, and we were able to see which strand was emphasized. All right. And so here we have one of the charts that we were able to create from the data that we produced, and this is the mortality of the tobacco budworms. Throughout these figures, you're going to see the 4 fields that we collected samples from compared to a benzoin colony. And basically what a benzoin colony is, it's just a neutral a neutral organization that has been keeping this insect for different generations, and so the insect hasn't been exposed to any type of extremes, meaning that it hasn't had the chance to develop any resistances that would kind of render it more resistant to the pesticide. So it's just like a very neutral control group that we would be able to compare our samples to. And so, as you can see here, there are different trends throughout each field, but you see that throughout the general trend throughout all the fields is that the susceptible population had a higher mortality, while the homogeneous resistant one was doing way better than both of the other ones. And so, next slide. We see this trend continue onto these graphs, which are the weight gain and loss throughout the process of being raised on the diet. And so what these two graphs do is they serve as a proxy to how the insect would do in the wild, since typically if the insect gains a lot of weight before maturing into a moth, then it would have a greater chance of surviving to reproduction after it's kind of matured. And so we see here from the insect gain, 75% gain graph, that the resistant strand has been doing way better than the susceptible strand, as the resistant strand has gained more weight. And we see the same thing over here, just opposite, where we see the susceptible strain losing the most weight, while the resistant one is losing the least weight. And so we finally come to this graph where we see the weight difference, and as you can see here, that the resistant one had the largest weight gain. So this just kind of took the final weight from the experiment minus the initial weight, leaving a positive value if you gain weight and a negative value if you lost weight. So you see that a large portion of the susceptible strand is in the negative area, while most of the resistant ones are in the positive area. However, you also do see some kind of variation, as sometimes the RS strand does better than the RR strand, or the SS does better than the RS strand. And I'll get more into that in a second, but that's just because there could be another mutation in there that we do not know. All right. And so, conclusions and findings, we found that the L1029H mutation was present in these insects, as it was targeting the sodium channels, and the resistance was confirmed within that strand. And again, further studies, we would go into finding this additional mutation that could be causing the resistance, where sometimes the SS strand does better than the RS strand, or the resistant one isn't as dominant as it should be. And so what we would do to go about this is that we would take samples that did really great from the SS, the blue strand, ones that gained the most weight, and compare those to those down here that lost the most weight, to be able to see what is the difference between the two and what exactly is causing that resistance. All right, and just a take-home slide from this is that pesticide research isn't necessarily what you think about whenever you go about your day eating the different vegetables that are grown or consuming the different products that are produced from agriculture, but it is very important to know that we should kind of take care of the fields and make sure and kind of like fund the research that is going into mass producing these different crops that are feeding a lot of people. And so I would just like to acknowledge Carolina Livery for offering transportation back and forth from NC State, the NCSM Foundation, NCSM, Dr. Shoemaker, Mr. Warren, Dr. Fred Gould, and Ms. Alice Bianchi. Thank you so much.
We have
Do you have any like information about what pesticides were used at the fields you collected?
What was that?
Do you have any information about the pesticides that were used at the fields you collected?
Typically, all the pesticides that were used in the fields that we collected was the permethrin pesticide. So that's just a common pesticide that's used right now since it was pretty functioning, but now farmers are starting to see that it's not working as great anymore, and that's just because the insects are becoming more resistant. And so our research focused on that specific pesticide since that pesticide, the permethrin pesticide, targets the sodium channel, and we saw that the general trend of the sodium channel not being affected, even though it was applied to the insects. So it was permethrin pesticide.
Was your field just like one big field that you collected in Durham, or did you go to multiple smaller fields?
Oh yeah, we went to multiple different ones. Like if you go back a few slides and the charts, you see that the different ones were like Rocky Mount, Reedsville, Cedar Grove, and there was another one. But yeah, we went all across North Carolina this summer to collect our samples. Yeah.
So, do you think similar findings would be found in different states, or do you think it's just a trend that happens in North Carolina?
It's probably very similar in different states since it's not necessarily an area type thing. It's just how the insects are responding to the pesticide and how they're kind of mutating against it. So it's probably a very similar trend.
Can you describe how you weigh the insects?
Wasn't that was mostly you?
Yeah, so you literally just take them up. So they have a lot of frass on them. That's a really nice word for poop. You clean them off, put them on a scale, hate your life a little bit, put them back in the cup, and then repeat. And so we had about 950 caterpillars, and to weigh all 950 took about 3.5 hours.
Wow. How did you even like count the little?
Yeah, so when they initially got the eggs off the buds, they had to sit there with a paintbrush and take them all off. Yeah.
Wow.
So I didn't do any of the counting they did.
It was like somewhat of a tedious process, but
Somewhat.
it gave us good numbers, and that's what matters.
How long did your collection take? The initial collection of
The collection of all the insects?
Yeah.
So we spent 5 weeks here for summer research over the summer, and for the most part, we collected most of them towards like the last 2 weeks because early on in the summer, it was like a really weird season. Like it was really cold. Like there was like a cold week somewhere in there, which threw a lot of farmers off. So early on, we couldn't really get that many samples because the kind of like what's the word? The cycle of how the insects reproduce was really thrown off. So it wasn't until like later in the summer when we were able to collect all of them. But yeah, it took about like the entire 5 weeks of summer.
That was so really hot.
It was really hot.